Okay, it looks to me like the numbers are evening out, so we might make a start. Just checking to see who's coming in. So yeah, thank you for joining these fundamentals of fluorescence microscopy webinars. My name is Paul McMillan. I'm the manager of the biological optical microscopy platform at the University of Melbourne. We run these webinars every year to try and increase the fundamental understanding of our researchers coming to use fluorescence microscopy applications. I did mention just in the chat just now, but we are recording these sessions and we will make them available on our webpage. I've just put a link to the webpage into the chat. We'll put that up again at the end. So this is a webinar series of three different webinars. We'll go through different content in each of those. If you have any questions at any point, feel free to use the Q&A chat function to post those questions. I will answer them at the end of the webinar. Otherwise, you can put your hand up and I can unmute you if you're brave enough to ask a question online. So today's lecture, we're going to look at fluorescence and fluorescence microscopy applications. I'm just going to put my camera off. So that's save a bit of that. Okay, so before we get started in today's webinar, I'd just like to acknowledge the traditional owners of the land on which we're meeting. For me in the Parkville campus here in Melbourne, that's the Wurundjeri Woiwurrung people of the Kulin Nation. I'd like to pay my respects to their elders past, present, and emerging, and any First Nations people who are joining us today for these webinars. The other thing that I guess is a little bit important is that although we run these webinars for the University of Melbourne researchers, they are widely available and have been advertised through the Light Microscopy Australia emailer. I am the president of LMA, and if light microscopy is your thing, then I would encourage you to become a member of LMA as well. So we are a special interest group of the Australian Microscopy and Microanalysis Society. LMA has 250 members plus across Australia. And here are some of the benefits you can get from being an LMA member. So we have travel bursaries for conferences, workshops, and lab visits. We're about to release a professional development scheme, which has $8,000 a year in it as well to our members. We run regular data analysis workshops. There's an email that goes out advertising upcoming events such as this, and also an annual image competition with cash prizes. So you use some of your science to get yourself some nice things. Our next big event that's coming up is the Australian Conference on Microscopy and Microanalysis. This will be in Sydney in February next year. That is 4 days of symposium, 4 days of conference that covers all of microscopy, so electron microscopy, X-rays, pretty much anything that has microscope applications. So 4 days of scientific talks, 2 days of workshops that will incorporate light microscopy applications, but also bioimage analysis applications as well. If you're interested, then you can go to microscopy.org.au to become a member or find out more information. You can also follow LMA on LinkedIn as well. So I mentioned that today is the first of a series of 3 different lectures. In today's lecture, we're going to cover fluorescence and fluorescence microscopy. This time next week, we'll cover sample preparation, some of the different experimental applications that are possible on optical microscopes, and a little bit around image ethics as well. And then in 2 weeks' time, my colleague Ellie Cho will be presenting the fundamentals of bioimage analysis as well. Hopefully this gives you a good fundamental understanding of the fluorescence microscopy workflow. Lecture 1, so we're going to break this up into 4 small sections. So I'm going to very quickly go over the BOMP platform. This is again primarily aimed at those of you who are in Melbourne. We're going to go through fluorescence, what it is and how it works. We'll then go through some examples of the different types of fluorescent proteins, tags, and dyes that we can use in experiments. And then we'll close out and we'll look at the fluorescence microscope and how a basic epifluorescence microscope differs to, say, a confocal microscope. And we'll look at some of the structural components of those systems through the light path of the microscope. So first of all, I just want to spend a couple of minutes just going through the BOMP platform and what we do. So this is what a common researcher looks like when they're coming in to come and speak to us for the first time. We've got this kind of brick wall that's sitting in front of you that has all these different things that you have to consider when you're planning and setting up and running and analyzing your microscopy data. And they're a little bit confused. What our job is as a platform is to try and break down this barrier for them. And what we're going to ideally do is take the bricks out, throw away the ones that we don't want, and use the ones that are important to build them their path through to the end of their project. And hopefully it's slightly less scary walk through the woods than it could have been without our assistance. So how do we do that? So the platform will try and help researchers as much as possible in the planning and experiment stage of their projects. So we'll talk to them about their experimental design, how they're going to prepare the sample. We'll teach them how to acquire the data, also how to process the data, and then finally how to quantify and extract numbers out of that data as well. And this is what the BOMP team looks like at the moment. So we currently have 7 team members spread across campus. We're supporting 21 microscopes, 7 analysis computers spread across the Parkville campus. I'll show you that in the next slide. These are some of the applications that are possible within the platform. Again, we'll talk about quite a few of those in next week's webinar. Also, we spend quite a lot of time on the image analysis side of things, and that's some of the softwares that we can support. If you're from Melbourne and you know what the Parkville campus looks like, then we are spread across 6 different locations. So from the By21 Institute in the west, our main lab and offices in the medical building. We also support equipment in the Peter Doherty Institute, School of Biosciences, School of Physics. And we're just setting up a node now at the newly established Aikenhead Centre for Medical Discovery, which is out in Fitzroy at St Vincent's Hospital campus. And yeah, this is the website you want to go to if you want to learn more information. So I mentioned that today's lecture is recorded. We've actually recorded and made available pretty much all of the workshops and seminars that we've ever run in the platforms. If you scan on that QR code, that will take you to the webpage that's at the bottom there. Here we have video links and PDFs of presentations that you can go to and go through that content if there's something there that might be of interest to your own research. So feel free to make use of that as much as possible. I'm just going to see there's a hand up. If you're putting your hand up, are you able to put in the Q&A what your response is? Because it's going to be very difficult for me to keep track of the hands being raised during the presentation. Thank you. Okay, so let's get started. So this is a fundamentals lecture. So we're going to start right at the very beginning, and we're going to look at fluorescence, what it is, how it works. And throughout this fundamentals lecture series, we are not anticipating any prior understanding. So we're going to start a really assuming that you have no understanding coming into these lectures. So fluorescence was first discovered by Sir George Gabriel Stokes in 1852 and was further defined by Alexander Jablonski much later on in 1933. Keep track on those names. They will come up over the next few slides. So Stokes and Jablonski, you can play your bingo card. They will come back up. So fluorescence can happen to a molecule when it undergoes this three-step process. So first of all, it will absorb the energy from a photon of light. That then transitions that molecule into what we call an excited state. And then finally, we have the emission, where that energy again is released back out from that molecule again as a photon of light. These molecules can come in lots of different shapes and sizes, and depending on the structure of them, this will change the characteristics of the molecules quite a bit. So this is something that we make use of, potentially for doing things like multi-channel, multi-stain imaging. And we'll go through over the next few slides what some of those characteristics will be. Before we do that, what we want to do again is look at that three-step transition of fluorescence. And this image on the left-hand side here, that's our molecule that we're looking at. On the right, this is the Jablonski diagram. So I told you before that Jablonski would come back up. Here, what we're looking at is the energy levels of the electrons within our molecule that we're interested in. So to start with, these molecules are at what we call the ground state. Here, the energy in the electrons is at the lowest state. Once we go through that first step, which is the absorption of the energy from the photon, these molecules go into what we call an excited state. So the energy in the electrons has increased. They will stay in that excited state for a very short period of time before losing some energy and reducing down to this minimally excited state. It's what we call the excited lifetime. I'll talk over the next couple of webinars around fluorescence lifetime as well. So here electrons will come down still in an excited state, but at a minimal point, before then reducing back down to the ground state. This is the point at which we have emission of that energy back out again as a photon of light. One of the nice things about fluorescence is that these molecules can undergo this process multiple times. So you don't just have one molecule, one photon being released. They can undergo multiple rounds of fluorescence, which is great for us. We get lots of signal from a few molecules. One of the downsides we might encounter is if we put too much light on, then we can get a physical breakage in the molecule, which changes the characteristics and causes them to no longer fluoresce in the wavelengths that we're wanting them to. This is what we call photobleaching. We don't want to, ideally when we're acquiring, we don't want to damage the fluorophores that we have there, so we can still get that cycle of fluorescence. So that's one thing to bear in mind when you are setting up your experiments. Again, if we go back to the Jablonski diagram here, so in this case, I've color-coded it by wavelength. So this molecule here, which is probably something like a green fluorescent protein, for example, we need to excite that with a photon with a blue wavelength, so something in the kind of high 400s nanometer range. So the electrons in this molecule will go from the ground state up to the excited state. I mentioned that they lose a little bit of energy there. Normally that's in the form of heat before returning back down to the ground state via emission. This loss of energy here becomes something that's really important for the microscopes. And as we go through how the microscopes are set up, hopefully that will be a little bit more obvious to you. The wavelengths at which excitation and emission can occur change for each of the molecules. So each molecule will have slightly different what we call excitation and emission spectra. So in this case, we have an example here for enhanced green fluorescent protein. You can see the excitation spectra in blue here and the emission spectra in green. You see they have very different shapes. We'll also notice there's a difference between the excitation maximum and the emission maximum. This is what we call the Stokes shift. And this is due I just go back to that loss of energy between the two transitions. And again, each molecule has a slightly different size of Stokes shift on there. So these are some of the key characteristics we have to think about when we're first designing our experiments in terms of what fluorophores we can use. So let's go a little bit more deeply and go one by one into these. So this is the excitation spectrum. So excitation can occur anywhere across this range, but will happen most commonly, most efficiently at what we call the excitation maximum. So if you buy a new dye, often you'll see excitation and emission maximum being referenced there. That's, I guess, the kind of minimal information you need to know about your dye. I would be trying to find the spectra, if you can, to try and help out with working out how to handle these samples. If we go to the emission spectra, again, emission can occur anywhere across this spectrum, but again, will happen most commonly at what we call the emission maximum, which again is the top of that curve there. Sorry, my pointer is for some reason being very laggy, which is why I'm not pointing it very well. One key thing when we're starting to set up our experiments is that we want to make sure that we're exciting our sample as efficiently as possible, right? So what this figure here shows you is that if we excite our molecule with the photons at the wavelength A, which is about what, 540 nanometers or something, we're going to get the most number of photons released from that molecule. If we excite down here, say at the 500 nanometer range at this position C, then we're going to reduce the number of photons that we're getting out. So we'll still get fluorescence, it's just not as efficient as it could be. Again, if you excite down here at 440 nanometers, then again, you're going to get some light out, but not very much. One thing you will notice is the shape of the curve on the emission side does not change. It's just the intensity of what's being released. So how efficiently are we exciting that molecule to produce photons to come back out. So the wavelength of what we're emitting doesn't change based on what we're exciting on this side. So I mentioned each different molecule has very different setups, and this becomes really important when you're planning your experiments to start with. So say, for example, something here that's got a fairly narrow Stokes shift. If you look at this example on the bottom left-hand side here, you can see there's a massive Stokes shift between your excitation and your emission there. So again, really important things to understand when you're first planning your experiments, making sure you've got the correct microscope set up for what you're trying to do. All right, so to summarize what we've covered so far, again, we're looking here over the wavelengths. So this is our excitation spectra. So again, excitation can occur anywhere across this spectrum, but will happen most commonly, most efficiently at the excitation maximum. This produces an increase in the energy of the electrons in the molecule. So this becomes into an excited state. We will lose some of that energy during the time it's in the excited state. That goes off as heat. That's what causes this Stokes shift, this shift to the right to a longer wavelength for the emission. And then finally, those photons are emitted back out from the molecule when we go from the excited state, down to the ground state. And again, that will happen anywhere across this emission spectrum, but again, most efficiently, most commonly at the emission maximum. All right, so what I'm going to try and do, if I can set it up correctly, is we have some questions that I want to put up just to see if you have understood what we've been going through on the fluorescence. So I would encourage you to spend some time filling in those polls. Hopefully you guys can see them. Okay, everyone, hopefully everyone's had a chance there to get through the poll. So the first question is, what is the correct order of events in fluorescence? So the answer there is, first of all, absorption, followed by excitation, and then emission, which looks like most people have got. Question 2 is, what best describes Stokes shift? The correct answer there, which again most people have got, is heat loss that results in an emission of a longer wavelength photon. So you remember the emitted wavelength is longer than the excited wavelength, lower energy, longer wavelength. And then finally, what should you match your illumination wavelength to? Again, that should be to your excitation maximum. Ideally, we want to match that wavelength that we're exciting the sample with on the system to the excitation maximum of the sample. I just realized I stopped a couple of slides early, assuming my summary would be the last one. So yeah, just to go through again, so we've looked at 3 kind of main characteristics of fluorescence. That is the excitation wavelength, the emission wavelength, and the Stokes shift. But there are a lot more properties as well that each fluorophore has. So the extinction coefficient is the same, as I mentioned, as the absorption coefficient. We have the quantum yield, which is, I guess, the efficiency of converting a photon that's been absorbed into a photon that's been emitted. We have a measure of the brightness, which takes those 2 counters into effect, so extinction coefficient and the quantum yield. pKa can be a really important one if you're looking at things with this in acidic vesicles, for example. This is acid dissociation constant. So the molecule will only fluoresce above this value, which is the pH. We have photostability, so I mentioned around the photobleaching issues. So half time for photobleaching. And then I also briefly mentioned fluorescence lifetime. So it's the length of time the molecule stays in that excited state. All right, so we've been through the poll. So we'll move on to the second section, which is fluorescent proteins, dyes, and tags. As I say, if you have questions that pop into mind as we're going through, feel free to put them in the Q&A section. So, good place to start in terms of fluorescent tags is probably the one that most people will have heard of already, which is the green fluorescent protein. So this is what the structure looks like. It has this beta barrel shape. The fluorophore forms from 3 amino acids sitting in the middle of the fluorescent protein. This originally came from jellyfish. And who knew, if you do jellyfish research, you can get a Nobel Prize. So Shimomura identified the GFP protein in jellyfish initially. Martin Chalfie was one of the first scientists to use that to tag proteins of interest and track them within biological systems. And then Roger Chen really took that and increased the color palette of fluorescent proteins that are available to us. So why are fluorescent proteins useful to us? So the nice thing is, as I mentioned, is it's a genetically encoded sequence, so we can attach it to a protein of interest. So if we fuse the DNA sequences in frame with each other, then we can create I guess, a mutant protein that has a fluorescent protein attached to our protein of interest. And we can, depending on how we do that, fuse the GFP either at the C terminus of that protein or at the N terminus of that protein. And yeah, this requires genetic modification, so either transfection, electroporation, CRISPR, viral transduction, that type of thing. Some of the considerations we need to think about when we're doing this is that, lots of proteins have specific trafficking motifs. So if you're trying to get a protein to go into the mitochondria, then there is an N-terminal sequence which encodes for that. You don't want to put your GFP on the N-terminus then of that molecule. If you're trying to get your protein to go to, say, the endoplasmic reticulum, then there is a C-terminal motif which encodes for that. So you probably don't want to attach your GFP molecule to the C-terminal of those proteins. Other things to think about is secondary structure. So if there is a secondary structure available for your protein, does the N or C terminus that you're attaching the GFP molecule to, does that sit internal to the structure of the protein, or is that sitting outside where you could add a relatively large tag onto it? The other thing to think about, and this is what I've shown in the panel below, is the expression levels. Often we're using very strong promoters when we're expressing these proteins. Again, if you overexpress, you can change the behavior of your protein of interest. So ideally, what you want to do is minimize the expression level to a point where you can visualize it without perturbing the system too much. And the other big thing to think about is the size of this protein. So a GFP molecule is 27 kilodaltons in size, which is fine if your protein is much, much larger, but if you have a much smaller protein, then adding that tag in can, a bit like dragging a ball and chain around campus. I'm not going to go to the coffee shop that's a 5-minute walk away. I'm going to go to the one that's probably a bit closer to where I work. It's changing the behavior. So fluorescent proteins are now fairly well established across lots of different species, as we can see here. And I mentioned in one of the first slides that they form this beta barrel conformation. So this is a side view, lots of beta sheets with the chromophore sitting in the middle. This is the top-down view. So you're looking within the barrel, and again, you've got the chromophore sitting in the middle there, nice and protected from the environment around it. Chromophore itself, as I mentioned, is made up of these 3 amino acids. It takes time for this to develop. I mentioned Roger Chen got the Nobel Prize for really taking fluorescent proteins and expanding on the capabilities and the different things that we have available to us. So what he and others did was to go through, find different species, as sources for these fluorescent proteins, and then go through and really start to mutate and try and work out what different amino acids did in terms of the fluorescent characteristics. And you can see some of the examples down here of different fluorescent proteins and some of the different mutations that they have within the fluorescent protein structure. Being able to do this, we now have color palette. This is slide slightly old, which goes all the way from the blue wavelengths all the way out into those deep red wavelengths as well. So that allows us to, have a few more molecules tagged within our samples that we can visualize. Okay, so before we went through the fluorescence properties, we had all of these characteristics. If we're looking at fluorescent proteins, there are a couple here that we need to now take into account. So the first is the oligomeric state of those fluorescent proteins. Do they like to form as monomers? Do they prefer to bind as dimers and tetramers? Ideally, when you're looking at, again, protein trafficking or protein behavior, you ideally want a fluorescent protein that wants to form a monomer. There are lots of those out that will potentially bind together as dimers and tetramers, which again can affect the characteristics of your protein that you're looking at. Next thing is maturation. So because these are proteins, they are transcribed and translated. It takes time for those molecules to fold. It takes time for the chromophore to form as well, which only at that point can you create fluorescence. So we have to look at the maturation rate of how long it takes before you can get fluorescence out of the fluorescent proteins. So there's a lot of things to consider there. And there's a lot of different fluorescent proteins. One resource that I would point you to is this FP base, which is a central repository for fluorescent proteins. Here you can use this interactive charts to try and work out, which protein might work for my specific experiment. These are some of the different characteristics that you can plot on this chart. At the moment, I've just got excitation wavelength and emission wavelength plotted there, but you can vary what you put into that plot depending on what's important for your experiment. I mentioned one of the benefits of having multiple different fluorescent proteins available is that we can use those for doing co-staining experiments. Example here is, perhaps a CFP, a GFP, and an RFP co-stain. So you can have different compartments stained with different fluorescent proteins. This table here shows some of the trafficking signals that I was talking about before that might be useful for moving your fluorescent proteins into specific compartments within the cell. The other thing you can do if you have access to them and you're bored on a Friday afternoon is you could paint a nice little beach scene with different colored fluorescent proteins that are growing in bacteria. Grow those on a bacterial plane. All right, so that's kind of standard fluorescent proteins. One thing I wanted to look at is some other potential applications of fluorescent proteins. We'll go through 3 of those over the next few slides. Now we know that the fluorescent protein wants to form as a beta barrel, which is formed by lots of different beta sheets. In this split fluorescent protein, we have our protein of interest here on the left. That is tagged to one of the beta sheets from the fluorescent protein. Okay, so we will only get fluorescence when these 2 proteins come into contact. So your protein of interest with the single beta sheets, along with what we call the split fluorescent protein molecule. Once they interact, then you'll get fluorescence. It's a good way of showing, if you direct your split fluorescent protein to a specific organelle, you can tell when your protein of interest is getting to that organelle based on the timing of the fluorescence. Benefit here is that single beta sheet is only a 15 amino acid sequence, so it's a much smaller tag, allowing your protein to traffic a little bit more normally. Some other tools that people have used are things like photoconvertible or photoswitchable fluorescent proteins. In this case, we can control what emission wavelengths these molecules emit over. So one example here is, say, for example, the EOS fluorescent proteins. We can control using ultraviolet light and create a structural rearrangement within the molecule, which converts it from, emitting in the green wavelengths to emitting in the red wavelengths. And again, we can use that to track protein trafficking, for example, over time after we've done that transition. So very useful. Can also be used in some super resolution applications, which we'll find out about next week. One other application, which again we'll talk about more in depth next week, is FRET biosensors. So this is Forster resonance energy transfer. This is a way of looking at protein-protein interactions. So say we have 2 molecules here that we're interested to know when they're interacting. If we tag one molecule with one fluorescent protein, another molecule with a related fluorescent protein, we can look at the interactions between those 2 molecules. So in this case, where there's no interaction, we excite the cyan fluorescent protein with its correct excitation wavelength, and it will emit that energy out. So we're seeing mostly cyan signal here. When this complex forms, that brings these 2 molecules together. They need to sit within 10 nanometers of each other for this to occur. But if we excite the CFP molecule now, rather than emitting the correct wavelength, it actually transfers that energy to the YFP molecule, exciting that, and we can measure the release from the YFP molecule instead of the CFP molecule. So here you can see when we have those interactions happening, that we see a shift in the wavelengths that are being emitted to more to the 500 nanometer range and more emitted from the YFP molecule. So that was fluorescent proteins. And as I mentioned, one of the issues we have there is those tags can be quite large. So 27 kilodalton for a GFP molecule, for example. So how can we get around those problems? And one of the ways we can get around that is to use something called an epitope. tag. So we already kind of used that with the split GFP molecule where we have a smaller sequence that we're attaching. So some examples of these that you might have heard of before are like a MIC tag, a HA tag, V5 tag. These are very short amino acid sequences that we can use to detect these tags. Often we will need to use either an antibody-based or a dye-based approach to detect these tags. So like a MIC tag, a HA tag, V5 tag, we would be using antibodies. If you use something like a halo tag, for example, then we can use dyes which will bind specifically to that halo tag. Often with these epitope tags, these short sequences, you will find that they will be used as repeated sequences. So like if you see a 3x HA, then there's 3 copies of this tag running in a line in your protein sequence, which allows you to bind 3 sets of antibodies to that tag. All right, while we're talking about antibody labeling, let's quickly discuss a couple of approaches that we can do to do that. So the 2 main methods are divided into what we call direct immunofluorescence assays and indirect immunofluorescence assays. In the direct IFA method, we have an antibody which is detecting our protein of interest, this little green thing on the surface of this cell here. And in this case, this antibody has a fluorophore directly conjugated onto it. Okay, so when the antibody binds the protein, we'll see that as fluorescence from that, coming from that antibody. Nice thing here is it's really nice and specific. We have a single antibody and a single fluorophore. That is also the downside, that we don't have potential amplification of the signal. So for every potential molecule of the protein we're finding, we've got 1 fluorophore attached. We can improve this slightly using the indirect immunofluorescence assay approach. Now, it's not shown here, but again, here we would have an antibody detecting our proteins of interest, the little green things on the surface of the cell. In this case, we have what we call a secondary antibody here, which is raised to identify the primary antibody. And the benefit we have here is, again, not shown on this diagram, but we could have multiple secondaries binding to this primary antibody and therefore getting amplification of the signal, which makes our imaging slightly easier. Downside is, if you've got an extra layer there in terms of 2 sets of antibodies, you start to come across limitations in terms of organisms that they've been raised in. We have non-specific binding and things like that become a problem as well. In terms of the antibodies that we have to use, so we can have polyclonal antibodies where it's binding on multiple sites within a protein that we're interested in, or monoclonal antibodies, again, single sites on that protein, no amplification of the signal, but potentially a lot cleaner in terms of the binding. We have different isotypes that we can use of antibodies to detect the proteins. Each of the antibodies are specific to an animal that it's been raised in. So for example, rabbit or mouse, and we need to be careful, certainly with the indirect method, that we're not mixing those up. Lots of people now are moving into the nanobody space, generated by things like camels, sharks, alpacas, all generate much, much smaller immune proteins that we can use for detecting proteins. Sources, you can either, buy things that have already been generated by companies from like Abcam, for example, or you can commercially design your own antibodies. Homemade just doesn't mean do it at home unless you have some alpacas, but I imagine you probably need a license to do that. The nice thing with the antibody-based approach is we have a full range of fluorophores that are available to us. So we can go all the way from the low 400s all the way up into the 800s, 850s, probably now pushing into kind of 900 nanometer wavelengths that allows us to do lots of different multiplex imaging if we want to. The other thing that I would say, just as a word of warning, I already covered photo bleaching a little bit in the fluorescence section, but we occasionally have people coming and trying to use dyes like fluorescene, for example. Some of the older generation dyes are not very photostable. So I would be encouraging you as much as possible to try and use some of the newer generation dyes. So things like Alexa fluors are a lot more photostable than things like FITSI. Again, that's worthwhile trying to get information on specification sheets to work out whether or not that your dye that you plan to use is relatively stable. Often we find people trying to transfer experiments that worked on flow cytometry into microscopy. One of the downsides is flow cytometers, the sample flies past the laser very, very quickly, so you don't need to have high photostability, certainly not as much as you do in microscopy. One other approach I've mentioned a couple of times now is multiplexing. So standard, microscopy experiments, you can probably do 4 colors pretty easily. We can probably push to 5 and maybe 6 now with some of the newer confocal systems. But if you want to multiplex beyond that, then we need to take some different approaches. One approach to that is something called Opal. It's a kit which comes with a certain number of different colors here. In this case, we're still using primary and secondary antibodies, but instead of the secondary having a fluorophore attached, it has an enzyme attached called horseradish peroxidase. This enzyme starts this reaction here to activate fluorophores, which then become covalently attached to your sample. We can heat treat the sample to remove the antibodies and then come in with a new set of antibodies. And you can do that multiple times to build up a, for example, 7-color panel. One last thing that we might cover, I don't think many people use these because they tend to be quite expensive, but there are quantum dots that are available. These again exhibit fluorescent properties, not using the kind of traditional Jablonski diagram type transitions that we talked about earlier. They come as these different shell and core structures, and the different size of the nanodots, quantum dots, tunes the wavelength of the light that's being emitted. The other nice thing is you can coat these quantum dots with different antibodies or oligo attachments to bind specifically to proteins or DNA or RNA sequences that you might be interested in. So this is what the excitation and emission spectra look like for these. So you'll see they have very similar excitation properties, but the emission spectra again look fairly like standard fluorophores, fairly narrow emission spectra on those. One last thing to look at are probes and stains. Now this could probably be a lecture in itself. There are lots of these types of things available. These are, specific dyes that are maybe cell permeable and are taken up into the cell, specific compartments within the cell. So one example of that would be DAPI, for example, that most people know binds to nucleotides. So you can see them preferentially in the nucleus. We have, cell permeable stains, which will go into mitochondria based on the charge differential within the mitochondria. So lots of different compartments that have these dyes available to you if you want to stain specific compartments. Here again is an example. So we've got DAPI here, which is preferentially staining the nucleus. We've got a cell mask, deep red in this case, which is in the infrared range, which is preferentially marking the cell membrane around the outside of the cell. TMRM, that's tetramethylrhodamine. This will mark the mitochondria within the cell. And then we have an ER tracker here in green, which is going to stain the endoplasmic reticulum in the cell. One thing just to be aware of with these probes and stains is that sometimes they are compatible just with live cell or just with fixed. Sometimes they are compatible with both live cell and fixed applications. And again, something to be aware of when you first start those experiments, when you're in the experimental planning stage. All right, so that's the end of that section. So let me just bring up the poll. Find what I've done with my cursor. So we're going to launch fluorescent protein section. So I'll give you guys a couple of minutes to work through that and try and answer those questions as best you can. Okay, so let's work through those questions. So the first one is what best explains the below approaches? So fluorescent proteins should have been matched with genetically encoded and can be used in live and fixed samples. For direct immunofluorescence, which that looks like for that first one, most people got correct. For the second one, again, most people got this correct. Direct immunofluorescence requires an antibody conjugated to a fluorophore. Question 3 is indirect immunofluorescence. Again, most people got that correct. Requires a primary antibody and a secondary antibody. The secondary antibody is conjugated to a fluorophore. And then finally, epitope tag, which again, most people got correct, is genetically encoded that can only be detected by antibodies and dyes. The second question, which of the below are properties that are relevant only to fluorescent proteins? So not included in general fluorescence. In this case, we are looking at oligomeric states and maturation rate. So lifetime and brightness are also characteristics of standard fluorophores, so aren't specific to fluorescent proteins. All right, so yeah, it looks like most people did pretty well on those questions. So the next thing we're going to go through, and we'll go through in quite a bit of detail, is fluorescence microscopes. And I wanted to put this up as a little bit of a lighthearted thing, but often we will see in emails that people want to get trained on the fluorescence microscope. So a fluorescence microscope is what you will tend to use in a facility or in your work. We don't often have very many fluorescent microscopes unless you accidentally spill some of your stain onto them. I would ask you not to do that if you can do that. But yeah, we're looking at fluorescence microscopy, not fluorescent microscopes. So what are the different types of microscopes that are available? So your kind of common microscopes would be this widefield or epifluorescence microscope and confocal, and these are certainly the two that we're going to cover mostly today. These are both actually epifluorescence microscopes, so the excitation and the emission wavelengths are going through the same objective. How they work is slightly different. So in widefield microscopy, we're illuminating the entire field of view and capturing that back to either your eyepiece or to the camera. We have a couple of systems within the platform, so a high content imaging and a slide scan system that use this wide field approach, camera-based approach. If you're ever looking at fluorescence down your eyepiece, then you'll pretty much be looking at wide field illumination. On confocal microscopy, we still have the excitation light coming out of the objective and the emission light going back into the objective, but we're not illuminating the full field of view at one point. We're illuminating a very small spot, and we're building up an image by scanning that across. And again, we'll discuss over the next few slides the differences between wide field and confocal microscopy. There are other microscope layouts. So one that we have in our platform is our light sheet microscope, which is designed for looking at large tissue samples. In this case, our excitation light is coming from two objectives that sit either side of the sample, and our emitted light is then collected by another objective here. There are lots of different layouts of light sheet microscopes that kind of look slightly different to that. We won't really cover those in today's webinar. All right, so let's start with this wide field microscope. And this, if we were to take a chainsaw down the middle of a microscope, this is what it would look like. If you're going to do that, please do that on any of our systems. But it's a bit complicated and probably a bit much for most people to understand. What we're going to do is simplify it down into this diagram. And what we're going to do is we're going to follow the photons. Okay, so we're going to start here with the illumination, what the light source is. And historically, certainly, it's been to create a broad white light, which we can then tune to the specific excitation wavelength that we want for our sample. This is done by an excitation filter that sits in the front here. This entire thing is called a filter cube. So we've got white light that we're filtering out. In this case, this would probably be like a GFP filter cube. So it's tuned specifically for one molecule that we want to look at. Here, we're only allowing the blue wavelengths of light to come through. They're going to hit off this dichroic mirror, which directs it down to the objective, through the objective and into our sample. That excitation light is going to excite our fluorophores. We're then going to collect any light that's been emitted, which goes in all directions, but we're only collecting the light that comes back up through the objective. It can now transmit through that dichroic mirror because this mirror is specifically tuned just to reflect this wavelength, but to allow that longer wavelength emitted light to transmit through. We then have an emission filter as the last part of the filter cube, which will clean up the emitted light. So it's just the wavelength that we want to see either through the eyepiece or onto our detector, which is normally a camera. All right, so we're going to go through each one of these 5 different steps along this light path and go through in a little bit more detail. All right, so let's start with illumination. As I mentioned, historically, this has been to produce intense white light. This is normally bulb-based, so you may have seen, microscopes having mercury bulbs, xenon bulbs, metal halide bulbs. Again, as I say, the idea is to create intense white light that we can then filter down to the specific wavelengths that we're interested in. More recently, microscope companies have shifted to light-emitting diodes, where the light is a bit more tuned to a narrower band. The benefit that these have over the bulbs is lifetime. So an LED has a lifetime of, say, 20,000 hours. A mercury bulb you need to change after 200 hours. So if you go back to the lab and you see the little notebook sitting next to your microscope, it's asking you how long you used the system for. It's probably because they need to change the bulb after 200 hours if it's a mercury bulb. You don't want to be in the lab when one of those explodes. So here, we've got an example of those mercury arc bulbs creating white light. There are a few peaks across the different wavelengths, but essentially it's to produce intense white light versus a light-emitting diode that's, creating maybe a narrower band of a specific wavelength for exciting our samples. I mentioned the next thing it's going to hit is the filter cube, and the first bit in the filter cube is what we call an excitation filter. So its job is to take that white light and filter out everything that you don't want and only allow the wavelengths that you want to transmit through and excite your sample. So these will normally be what we're called a bandpass filter. So they're allowing a narrow band of wavelengths to go through and excite your sample. This again is what it would look like if you took a cut through that filter cube. So this is the incoming light. This is our excitation filter. If you remember, that light is filtered out. This dichroic is specially tuned to allow just the light that's come through to bounce off down to our objective. Our sample is going to get excited. We're collecting the emitted light coming back up. It can transmit through this dichroic because of that Stokes shift. It's now a longer wavelength. It can travel through. We hit an emission filter again to filter out anything other than the wavelengths that we're interested in to visualize. And then that goes up to the eyepiece or to the camera. So I mentioned light coming in through the excitation filter, off the dichroic mirror and down to the objective. So this is probably the most important part of your entire microscope. And I realize that normally for people, this is hiding under the stage or you can't really get in to see it. So I thought I would go through some of the information that's included on the objectives and tell you a little bit about what they are and why they're important. I would say for any of the bump microscopes, we have specification sheets that have all of this information available within that if you need it. So let's start at the very top. We've got the manufacturer here. Next information we've got here is plan APO. So these are aberration corrections on the objective. So plan in this case is a flat field correction. So how evenly in focus is your entire field of view. So on a poorly corrected lens, maybe the central part of your image is well in focus, but as you move away, it's a little bit more blurry. For the APO correction, this is what we call a chromatic aberration correction. This looks at how well focused are each of the wavelengths across the wavelengths. How well focused are they on that particular objective? So for example, a poorly corrected lens here, you might have a focus offset between each of your individual channels, which you would need to adjust for. In a better corrected lens, then you have less of those focus offsets. So if you imagine perhaps you're trying to do like co-localization analysis, if your green and your red channel have a 300 nanometer offset, then you're going to get very low co-localization between those two channels. That's the case. We have beads available that we can correct for those aberrations. Next thing down, which everyone tends to get hung up on, is the magnification. So this is the optical zoom value that this objective will give you. The number that's probably more important is the one that comes after that, which is the numerical aperture of that lens. And that gives you a readout of the resolution, and we'll talk about that in the next slide. After that, this lens we're showing here says oil. This is the immersion medium that objective is expecting. So it's requiring oil between the objective and the cover slip to get the best possible imaging quality out of that objective. You can have lots of different immersion mediums, for example, water, silicon, glycerol. If there is nothing written there, then it will be an air lens, which requires nothing between the objective and the cover slip for imaging. Next thing down, we have any specialized optical properties. So in this case, we've got DIC, which is a transmitted light contrast method. Another really important thing for us to look at is this number here. So this 0.17, that corresponds to the thickness of the cover slip that we're expecting. So this is in millimeters. That's 170 microns, which corresponds to a number 1.5 cover slip. Okay, so I guarantee you go back to your drawer, you'll find lots of number 1 cover slips sitting in there. If you're trying to do any imaging, especially deeper into a sample, or you're trying to do 3D imaging, I strongly recommend that you use a number 1.5 cover slip, whatever the objective that you're using is anticipating. Some objectives come with correction colors, so you can correct for this, but yeah, just make sure you're using the correct one. The final thing we're looking at here is WD 0.21. So this is the working distance of this objective. In this case, again, this is in millimeters. So the value is 210 microns. That means with this objective, you can only image 210 microns past the cover slip into your sample. Once you go beyond that, you're going to start lifting your sample up with the objective, which we want to try and avoid any damage to the front part of the lens. So we're going to work through some of those things, specifically around numerical aperture, cover slip thickness, and working distance. So I mentioned everyone gets fixated on the magnification. So when we're actually looking at the resolution of what's possible on this objective, it's actually the numerical aperture that's important, so not the magnification. So if we look at the equation, there's only 2 things we need to take into account. Here we've got equations for widefield and confocal. We've got the wavelength of the light that we're collecting and the numerical aperture of that lens. Okay, so this table shows us, I think, for the confocal data sets, actually maybe it's widefield, the effective resolution of these objectives. So if you have a objective with a NA of 1.4 and you're using oil there, then you can get theoretical resolution down to about 140 nanometers. In realistic terms, it's probably more like 200, 250 nanometers resolution. One important thing to bear in mind here is both our 63X and our 100X have a 1.4 NA. 1.4 NA. In this case, we would use the lower magnification lens as it's brighter, collects more light than the 100X objective. Okay, so here that's why you often don't see a 100X objective on a confocal system. So again, there's an equation there if you want to work that out. So this is lateral resolution in the XY direction. If we look at axial resolution, so in the Z direction, often this is 2 to 3 times worse than the lateral resolution. And again, here's the equation. We've got wavelength and we've got NA there. There's no magnification involved. The picture I'm showing underneath shows you what a point of light looks like in the microscope. So if we image it from above in the lateral direction, the XY direction, it looks like this. These rings that we see here are called airy disks. We'll come back to that when we're discussing confocals. And on the side here, these funny shaped things are what we call the point spread function. This is what it looks like in the Z direction. So you can see it's got that elongation. This is the way that the objectives work. That means that we have worse resolution in that Z direction. So here we've imaged the same field of view with 2 different objectives with different NAs. And you can see, hopefully, that you're getting more information in this higher numerical aperture lens than you are in the lower NA lens. Similarly, if you're doing things like co-localization, try and do that on a high numerical aperture, a high resolution lens. If you try and do co-localization analysis on something with a really low NA, then everything's going to look like it's co-localized, right? I mentioned this already. So numerical aperture is factor for brightness. So here, if we've got the same magnification, we select the higher NA lens for better resolution. If we have the same numerical aperture, then we're selecting the lower magnification lens for more light. Again, the equation is there if you want to work through that yourself. Next thing we want to look at is the working distance of the objective. And there's 2 bits that come into this. So I mentioned before that we want to try and image through those number 1.5 cover slips. They have a thickness of 170 microns. This is a section of one of our specification sheets on our microscopes. So here we've got what the cover slip thickness is. So these are all expecting that number 1.5 cover slip thickness, and they're giving us what the working distance is. Okay, so in this case, we've got, we're using the correct cover slip thickness. We can use the full working distance of each of these objectives to image into our sample. So on the 5X lens here, we can go 18 millimeters into the sample. If we want higher magnification, higher resolution, then we can only go 140 microns into the sample. So you can see up front, you know, if you've got tissue spheroids or you want to image a zebrafish, which are going to be a few millimeters, then you can probably only use the lower magnification lenses. If you've got cells sitting on a cover slip that you want to image at really high resolution, then you can do that on the 63X objective. Commonly, we get people coming and they're trying to image, maybe they bring their samples in like a 6-well microtiter plate, cell culture plate, or they are accidentally trying to image through the cover slip, slide thickness. They are, you know, often 1 to 2 millimeters thick. In this case, we can't even focus through and get into our samples on these high magnification objectives. So the working distance isn't sufficient for us to get through. You would only be able to image those on 5 or 10X objectives. We do have a couple of systems with long working distance lenses that can help for this. But yeah, try not to image through that thicker plastic or thicker glass. Try and mark your samples appropriately on the correct cover slip. So I mentioned that the objectives are probably the most important part of the microscope, and therefore we need your help to try and help sure that they're maintained properly. So I mentioned there are lenses normally have an NA less than 1, which don't need anything between the objective and the cover slip. These are what we call air lenses. In this case, we're trying to avoid contact with those immersion oils. If you're ever on an air lens, a low magnification lens, and you're struggling to find focus, or the samples look really fuzzy, it may be that someone before you has accidentally contaminated it with one of the immersion oils. If this is the case on a bond system, please let us know and we will come and clean that for you. Sometimes it can be a bit difficult to get that out. When it comes to the immersion lenses, then we need your help, and we ask you to clean those at the end of every session. So in the bond systems, what we ask you to do is to do a 3-step process. So we provide lens tissue for the researchers. Please don't use Kim wipes or standard tissues. They're far too abrasive for the objectives. What we ask you to do is take one strip of the lens tissue and gently kind of dab it over the top of the lens. And you can see the first dab here, we've got lots of oil. By the time we go down, it's coming less and less and less. We've taken the majority off. For the second step, take a fresh piece of lens tissue. We normally use 80% ethanol here, soak it in 80% ethanol. And in this case, I'm just dropping that on top of the lens, and I'm slowly pulling it across in one direction. So I'm not applying any downward pressure. I'm just using the wicking of the solution to kind of clean that off. Finally, third step, third piece of lens tissue, very gently around the outside part of the objective, not across this central glass part. Please soak up any excess ethanol or oil that hasn't already been picked up. You can also try down the side of the objectives as well. Sometimes people put on too much and that overflows down the side of the objective. But yeah, really important for you to try and help us take care of those objectives. All right, so if we remember we're a photon, so we've gone through excitation, we've gone through the excitation filter, we've hit the dichroic, we've gone through the objective, we've now excited the fluorophore in our sample, we're collecting the emitted light that's gone back up through the objective, through the dichroic, and the last thing it's going to hit before it gets to our eyes or the camera is the emission filter in the filter cube. There are lots of different types of emission filters that we can include. So things like short pass, long pass, and band pass. I've got a couple examples here. So for this example, this is a long pass 520. So it's going to let anything that's longer than 520 nanometers of light transmit through. And then we can visualize that on the microscope. If this was a long pass 520, then it would only allow wavelengths that are shorter than 520 nanometers to transmit through. And band pass again is going to allow a very narrow band of light to go through. So in this case, we have a dual band pass filter, which allows both green and red light to go through. In this case, we'd be using LEDs to illuminate the sample and illuminate just the green stain at one point, turn that off and turn on the LED to excite the red stain. Again, this is an excerpt of our specification sheets, which again shows you all of the components of the filter cube. So here we've got the excitation filter cube. Again, this is a band pass. Our dichroic here, these are long pass filters. And then finally on the emission side, these are again band pass filters. So what we want to try and do, this is spectral viewer from Thermo Fisher. These are 4 stains that we're including here. The dotted line are our excitation filter spectra, and the solid lines are our emission spectra. What we want to try and do is just make sure that these emission bands actually match with where our light is being emitted, right? So DAPI is probably okay, GFP is pretty good, RFP, maybe we're missing this shoulder a little bit, CY5, we're maybe missing this shoulder a little bit in terms of collecting that light. So again, something to consider when you're designing your experiments. what filters does my system have that to appropriately detect the fluorophores that I'm using. Last thing we'll look at is the digital cameras. So I mentioned wide field, we're illuminating the entire field of view. We're reading that out onto a camera in one shot. And there are lots of different types of cameras that are available. The different types that you would use really depend on the different applications that you want to use within our platform. We have one system with electron multiplying charge coupled devices. This is ideally set up for single molecule imaging. So so fairly faint samples. We have other systems set up with scientific CMOS cameras, which again have very fast readout speeds, so would be very useful for live cell imaging type applications. And some of the things for us to think about are, the quantum efficiency of the camera, and we're up now with these cameras up to kind of 95% quantum efficiency, so very, very efficient in terms of converting a photon that's hitting the camera into an electronic signal. We'll cover, I think later on in this talk, the importance of the pixel dimensions in our image as well. Another really important thing is monochrome versus color cameras. So if you're doing fluorescence-based readouts, you want to do that on a monochrome camera as much as possible. If you're doing colorimetric readouts, then you can do that on a color camera. And bit depth we'll also cover in the next few slides as well. All right, so that was a kind of fly through a wide field fluorescence microscope. What I want is to cover in the last section that we'll go through is the differences between that basic wide field fluorescence microscope and confocal microscopes. And again, we'll fly through the light path and discuss those changes as we go through. One thing I did want to highlight is that these basic fluorescence microscopes are still very powerful, so they're a very good way of collecting light. I often tell our researchers to try and optimize their stains as much as possible on a basic fluorescence microscope. It's a lot faster and a lot cheaper to do that than to try and use a confocal for optimizing your stains. You can still get really fantastic data off a very basic fluorescence microscope. You would potentially come to a confocal system when you want to get that, higher resolution, higher quality image. Here you can see we've got really nice signal to noise. The signal's quite strong. The background's pretty black. We're also getting optical sectioning here, so we're getting rid of any out of focus fuzz that we've got in the basic fluorescence microscope. We'll go through how a confocal can do that over the next few slides. So what's different between the confocal microscope and the wide field microscope? So we still have a light source here. In this case, we're using laser-based illumination. One thing which isn't shown on this diagram is a scanner. So I mentioned we're using that laser and scanning it across and building up our image over time. So I'll show you how that works. The objective is the same. One other thing that we have, which is different, is this pinhole here, which sits directly in front of our detector. And this is the thing that's blocking that out of focus light from other focal planes. The detector is very different, so it's not a camera-based detector. Mostly it's a point detector. And then, just the fact that we are using a scanner to build up the image over time means it's a computer-generated image rather than reading out into a digital camera. So we'll go through each of these in turn and discuss those. So laser illumination. So the nice thing here is the monochromatic, so single wavelength. So we often don't need excitation filters here. One of the good things here is there are narrow parallel beams, so they're collimated. The downside is, we can't illuminate the entire field of view in one shot. One of the downsides is it can be quite intense and powerful light that we're putting on. So again, we've got to be careful in terms of photo bleaching, phototoxicity of our sample. So here's the example of maybe an argon laser showing excitation probably around the 490 mark. We're creating just that blue light that we would use to excite something like a GFP molecule. One of the really cool things now is that we have lasers available to us that can span all the way from kind of 355 nanometers all the way out up to in kind of the close to the 800 nanometer range now. Whereas before we would have stopped probably around this 640 mark. We can now push more into the near infrared zone. That gives us, at least a couple more dyes that we can add into our panels without too much effort. Here, again, we've got our excitation and our emission spectra. Here we've got the Alexa Fluor 546. We discussed already the importance of matching the laser line you're using to excite your sample with the excitation maximum of your molecule. This is just another way of showing you what I already showed you. If we change the excitation wavelength, then we're decreasing the amount of light that we've got coming off. Okay, so again, the shape isn't changed, it's just the intensity of the light that's coming off. I mentioned that rather than illuminating the full field of view, we're scanning that intense beam across our sample and assigning intensity to pixels as we scan across. This scanning is performed using a couple of mirrors. So you may have heard of something called a galvo mirror. Sometimes there are mirrors called resonance scanners as well. So a galvo mirror, you can change the speed that you can scan across. On a resonance scanner, it tends to be set at a certain speed. What we're doing here is using at least two sets of mirrors, and these position the beam across our sample. So we can point the beam at a position in X and Y on our sample. So we can use that with those two mirrors in tandem to scan across our sample. So here in this example, we've got what we call a bidirectional scan. So it's going that first line of pixels from left to right, second line of pixels from right to left, third line of pixels left to right. I guess it's taking that time to scan and build up our image. The other important thing is that not just the excitation light goes through that, but the emission light. So that's an error, that should say emission light returns back through these mirrors as well, and that allows us to have a point detector at the end rather than a camera. Okay, so this is what we call descanning. The emitted light comes back through those mirrors. As I mentioned, on the galvo scanners, we can change the speeds. So if we move that scanner really quickly, that means that we're not sitting over each of the areas for very long. We're not getting very many photons back. That can result in quite noisy, rough data. As we slow that down, you can see that it cleans that data up. If we go really slowly, we're going to get really nice data, but we've also potentially bleached our sample as well here. So we've got lower intensity than we did over here. So we need to try and find a sweet spot in terms of how quickly we scan across that sample. Next thing we discussed was the pinhole, and as I mentioned, that's the thing that gives us the optical sectioning capability on the microscopes. So it sits directly in front of the detector. This is widefield, so same field of view imaged. In this case, we don't have a pinhole. We're reading out directly to a camera. We can see light that's coming from above and below our current focal plane, right? So we get lots of bright signal, but it's a little bit hazy, not very well focused. We go to a confocal system and we take the pinhole and we start to narrow it down, and you can see it starts to increase that optical sectioning capability. Question we often get is, at what point do we stop, right? At one point, you're going to close that pinhole, you're not going to get any light coming through. Is there an optimal place for setting that? So this is, again, just to show you how that pinhole works. So the dotted line is our current focal plane. Each of these shows a different example of light coming from a different position within the sample. And it's only when the fluorophore sits in our current focal plane that it can actually transmit through that pinhole. If it comes from below or above, you can see it's blocked out by that pinhole value there. So yeah, we're only going to see light that's at our current focal plane that can transmit through the pinhole. So let's go back to, what size should the pinhole be? And I showed you earlier on these airy discs. So this is what a point of light looks like on a microscope. If we image a sub-resolution fluorescent bead or something, we will get these intensity values. What we're trying to do with the pinhole is to match this central disc here. And what this is called is the airy unit. So if you come to confocal and you see something like 1 AU or airy 1, this is corresponding to that airy disk. So we're trying to just get the most intense information from that point of light. This works not just in XY, but also in the Z direction as well. So you remember the point spread function I showed you earlier on. That will also work in the Z direction. So again, we'll block out a lot of this out of focus light from the system as well. That's what's giving us this nice, clean optical section. Finally, I mentioned we're going through descanning through the scanner. The light can only cross over through the pinhole. So what we have at the end is a point detector, not a camera-based system for most confocals. Again, it's got a similar job, so it's detecting photons and converting that into an electronic signal. How it does that is very different depending on the different detectors. Sometimes we have simple signal amplification steps where we're adjusting what we call gain. So amplifying up the signal. Some problems we might come across is if we increase the gain, we're often increasing the noise of the detector as well. And yeah, we can work in two different modes. So integration mode where we're collecting signal and associating that with pinholes, or photon counting mode where we're trying to convert every photon that hits the detector into a photon count coming out. So we're a little bit more directly quantifying in that case. There are lots of different types of confocal detectors, way too many to go into during this lecture. But again, during the training, certainly within our platform, we would take you through and explain which ones you would need for your specific experiments. You'll see they all have different pros and cons. So for example, the gas detectors are really highly sensitive in these shorter wavelengths, so 600 out to kind of 700 nanometers. Once you go past that, you'll find that different detectors are required for that in different near-infrared wavelengths. But again, we would take you through as part of the training in terms of trying to set that up. I mentioned around the issue with gain, amplifying up your signal. So here maybe we've got a very faint signal that we've had to either image to reduce photo bleaching with not enough laser, so our signal to noise isn't very good, or we've had to amplify up using gain. That's amplified the noise in the image. One way we can get around this is to do something called averaging or accumulation. In averaging, we're going to take, in this case, 2 or 4 images and average the intensity at each of the pixels in the image across those 4 images. So it takes 4 times longer, but it can increase your signal to noise and therefore give you much better image quality. The other thing that we need to look at is how we're acquiring the data. So to make sure that we're not creating problems for ourselves when we come to analyze. So if we wanted to, take a representative image of this mountain to show how high it is, which of the 3 images do you think gives you the most information? So it is the one that's got the foreground in here? Is it the one that's zoomed in just on the mountain? Or is it the one that's zoomed in on the mountain that we can't even see the top? So hopefully you don't think it's this one. Out of these 3 images, probably this one is the best, but it gives you as close to as much of the information as possible. Ideally, we'd want to zoom out until we can see sea level and we could understand, how high it is above sea level. We need to do the same thing when we're setting up our microscope experiments. So here, if we look down, look up here, we've got some confocal microscope images that we've taken. This is with what we call the green lookup table. Underneath this, we've got a range indicator in there. What the range indicator does is shows 0 intensity pixels as blue. It then goes from black to white through grayscale, and finally shows saturated pixels in red. So in our first image here, we've used probably about 50% of the dynamic range of that detector. So that's probably okay, but we can probably get away with more, right? You want to use as much of the dynamic range as you can. So this is probably fairly optimal. Here, maybe our laser was too low or our gain settings were too low. We haven't amplified up the signal enough. In this example, we've got, we've gone too high with the gain or the laser, and actually, we've clipped off the top of that mountain. We can't tell what the intensity values are. We can't quantify any of the pixels that are in red within this region. In the other example, we've gone even worse. We've played around with a thing called offset, which is clipping the lower information out. So here we don't know the intensity at the top, and we don't know the intensity at the bottom. So this, again, these two options I would say are probably not what we want to do. This is optimal, and this one is okay. I mentioned that the system is generating the images. So confocal, we're scanning across, assigning intensities into pixels as we go. So it takes time to acquire this data. At the end, we see it as an image, but essentially it is an Excel spreadsheet of intensity values, right? So each pixel has its own intensity value, and we use that to build up the image that we can see on the screen. So last few slides that I want to go through is we will discuss digital imaging and some of the important things around pixelation when we're capturing digital images. So I mentioned we're seeing as an image on the screen, but essentially it's a numerical array of pixels. And each pixel has two key characteristics about it. So first of all, a size or a corresponding size within your sample. So in this example, this pixel is 100 nanometers by 100 nanometers within your sample. It also has an intensity value. So in this case, it's 108 intensity values. The range of this, or gray level values, the range of this is defined by something called the bit depth, which will I don't think we're covering in this bit, but certainly in terms of the size of the pixels, we'll talk a little bit about the sampling resolution. So our problem is when we're digitizing this, we're fixing in those pixel sizes at the time of acquisition. So the problem, as I mentioned, is that when we're doing that, we're potentially limiting the resolution of the image that we're creating. So therefore, the pixel size we're acquiring the data with becomes crucial. So the rule of thumb, and I'll go into the theory behind this in a couple of slides' time, is that your pixel size should be about 3 times smaller than the resolution you acquire. So if you're trying to resolve an object, it should be at least 3 by 3 pixels for you to be able to do that. And the example down here shows, very small pixel values with increased pixel values. And hopefully you can see the decreased image quality that we're getting as we go through this. So one little thought exercise for you to think about is that you've been looking down your microscope and you can see some objects there and you want to count those objects. What we want to do now is capture that data into a microscope image, and we're going to set a number of pixels there to do that. So hopefully you would agree with me that on this 3 by 2 pixels, we can't see any of the information that we were able to see down the eyepiece. If I increase the number of pixels, then we can probably start to see some of the objects there, but I don't think you could probably hand on heart say that you could definitely resolve and count each of those objects. By the time we get to 48 by 32 pixels, we can probably fairly comfortably say that we can see 7 different objects within that image, right? So if we're only interested in counting, then that is probably fine. What if we want to differentiate the different types of objects that we have in the image? So if we go to 96 by 64, then we might be able to work out that actually one of the objects here is slightly different, has a slightly different classification to some of those other objects. By the time we get to 192 by 128 pixels, we can see that we've got 6 wombats here and 1 brown pig in our image. But if we were wanting to maybe count, say, the number of ears or the number of eyes or the number of nostrils in that image, we probably don't have enough resolution to do that. By the time you get up to this value, then we're probably a little bit more confident in terms of being able to count those different objects. So the take-home message I'm trying to transmit here is that we need to think up front when we're acquiring the data what we will need to analyze at the end on that data. So it's worthwhile thinking about it up front, making sure you've captured the data correctly. Don't get to the end of your experiment and then work out you haven't acquired it properly. Now, I mentioned that there was a bit of mathematics behind this. Obviously, there's the sense check of whether you can see it yourself. If you want to err on the side of caution, then you can use something called Nyquist-Shannon sampling. There's a calculator here. Often you will find in your confocal software that there is an optimal button or a Nyquist button, which will do this calculation for you. This is telling that your pixel dimensions should be 2.3 times smaller than your theoretical resolution. So if we go back to this table example again, and we're in the 1.4 NA63X objective, again, this is a theoretical value. It's suggesting something like 60 nanometers pixel sizes. This is to get the best possible resolution you can get out of that microscope. So on the confocal, you could go down to 20 nanometer pixel sizes, but you're not going to get increased resolution from that. You're just oversampling. The other key thing to think about is that you should only use this if you actually need that level of resolution. So if you're trying to count the number of nuclei in an image, you don't need a 60 nanometer pixel size when your nuclei are way, way bigger than that. So in this case, this is just to get the best possible resolution you can off the microscope without it taking forever to acquire the data. All right, so we've gone through quite a bit on the fluorescence microscopes. Let me bring up the last poll for the day. Find you. We'll give you guys a couple of minutes to go through those and answer those. And I think we'll move on. We've hit over 50% participation there, which is good. You guys didn't fall asleep during the seminar. So the first question asks, please match the below attributes of an objective to the relevant effect. So numerical aperture, which I can see most of you got right, is attribute of image resolution. For planar apo, again, majority of you got it right. That's aberration correction. Magnification is the image field of view, which again, most of you got right. And working distance is the depth of the sample that can be imaged past the cover slip, assuming you're using the correct thickness of cover slip. Which of the following can be included in a filter cube? Select all that apply. So out of the 5 that are there, the 3 components of the filter cube are the excitation filter, the dichroic mirror, and the emission filter. And then finally, which of the following are characteristics of a pixel in a digital image? The 2 that correspond there are the size and the intensity value. So certainly there is no volume in a pixel, that's a voxel. And the resolution, it depends on the number of pixels that we include within the image. All right, so yeah, hopefully you guys found that interesting. So just to go back through what we have covered so far. So we gave a brief overview of the Blanc platform. We've covered fluorescence. We've covered fluorescence proteins and tags and dyes. And we've covered the structure of both a fluorescence microscope and a confocal microscope. I would remind you that next week's lecture is going to cover some general guidelines on sample preparation. We're going to go quite in depth into some of the experimental applications that we can do on advanced optical microscopes, and touch a little bit around image ethics in terms of modifications that we can make to digital data. In 2 weeks' time, my colleague will go through the fundamentals of bioimage analysis, which is an overview of the workflow of performing bioimage analysis. I'm going to put these QR codes up if you're interested to join our mailing list. We advertise upcoming events, primarily in Melbourne, but some of them are virtual. We also have a link there to our social media accounts if you want to follow us on those. Otherwise, if you have any questions, then feel free to stick them in the Q&A and I will try and answer them live. If you want to stick your hand up and ask it live as well, then let me know.